Okay, good afternoon, everybody, and welcome to our bi-weekly seminars organized by the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics and the Global Alliance for Immune Prediction and Intervention. This webinar is co-organized by myself and James Glazier, as well as Jim Sluka and Lorenzo. If you have any questions or comments, suggestions for speakers for this webinar, please feel free to contact any one of us here. We also have several channels, social media, and other media where we share different announcements as well as these presentations. In particular, our YouTube channel has all of these webinars archived, so you're welcome to check them out. And you can also please help us share more information about the working group and so forth. We have a couple of announcements first. So there are a number of funding opportunities with respect to medical digital twins. Here is one that's an interagency collaborative opportunity by NSF, FDA, and NIH. There is also a number of upcoming webinars hosted by the National Academies of Science on digital twins panels. May 9, May 16th, 23rd, and 30th. Links are available here. Feel free to register. We also share faculty positions or job openings related to digital twins and multiscale modeling. There are several faculty positions at the University of Florida in the Computational Systems Medicine and the Laboratory for Systems Medicine. Feel free to contact Reinhard Laubenbacher directly if you have any questions. Otherwise, you can check out the positions at this link. Another announcement on a workshop, a CompuCell3D workshop that will happen on July 20th through August 3rd. There will also be virtual tissue modeling hackathon during August 9th through the 11th. If you have any questions, James Glazier will be happy to answer them. Finally, we also have, we feature publications by the group and related publications. Here are two more recent papers on relevant to, again, medical digital twins. If you have any papers that you have published and would like to feature, please contact us. Contact Jim Sluka at jsluka@iu.edu, and we'll be happy to post them on our website. Do we have any other announcements? Anybody would like to add anything? Okay. We have, as you can see, we have a few gaps in webinar speakers coming up here. If you have any suggestions for speakers, please send them our way. If you would like to nominate yourselves as speakers, we're happy to also have you. And at any time, if you have any suggestions, if you read a paper or hear a talk, if a name comes up to you that you think would be an appropriate speaker for us, please send them our way. We like to increase the diversity of our speakers, the topics, and so forth. Okay, with that, it's my pleasure to welcome Randy Heiland from Indiana University, who will talk about open virtual tissues, a standardized ecosystem for virtual tissue simulation. Randy, welcome, and feel free to share your screen. I'll stop sharing mine.
Well, thank you, everyone, for taking time out of your busy days. My name is Randy Heiland. I'm a research associate here at IU. And I'm going to be talking to you about this OpenVT project, which has been underway for 6 months or so now, longer than that, I guess. So there's the name of if I consider the core team, Bart, Pete, Kyle, Michael, Hayden, and the PIs, of course. But more importantly is we consider this to be a larger team effort, and that includes developers of any multicellular modeling frameworks. So the link at the bottom is for these slides. So you can go grab them in case I don't finish on time or you just want to peruse them later. So we have an OpenVT GitHub organization, and in there's a repository just called openvt-edu, and you'll see the slides folder under that. So the outline of my talk, which looks a little wordy and time-consuming, we'll see how it goes. I wanted to briefly discuss this NSF I-Core program, which we all went through at the beginning of this project. And then I'm going to dive right into what are some fundamental multicellular modeling approaches and some of the existing frameworks that are out there and the features of those frameworks. The bulk of this talk, and quite honestly, the project was funded to primarily focus on cellular pots and center-based approaches to modeling. So that's what I will do in these slides for the most part. And since standards is in the title of the funded project, we will indeed be touching on those, even though I tend to use quotes around the word standards because it's kind of a loaded word, at least for me. And then I'll share some early efforts that we've accomplished just converting between these frameworks, PhysiCell and CompuCell. We will wrap it up sharing some of the reference models that we've all agreed on, at least to date. I suppose there's some room for flexibility still, but we'll share those. And then the final topic will be on reproducibility. Since the goal of this is to be able to use multiple frameworks, then it makes sense to talk about, being able to reproduce a model among multiple frameworks. So that'll be the last topic. So this project, I'm going to see if I can get rid of this thing. Maybe not. There we go. Was part of this POSE project through the NSF. And these awards were given out around September of last year, and there were roughly 19 or 20. You can go to that link there and track down all of these pretty interesting sounding projects, actually. But the focus is on projects that are developing open source software. And not surprisingly, the NSF wants to strongly encourage making projects like these sustainable, right? The NSF is not in the business to give out grants just willy-nilly. They would like to see these projects become sustainable. So the awards were in September. In December, all of the awardees were notified that they would need to participate in this NSF I-Core program starting in January. So that was a bit of a shock to the system, given that, everyone had turned in their project proposals with budgets and timelines. And then the NSF came back and said this. What's going on here? Oops. Yeah. So what is this I-Core program? You may ask if you haven't participated in it yet. I actually was made aware of it some years ago when I was working on the NSF Science Gateways projects, for any of you who know about that, but I had never participated in it before. But it's basically an entrepreneurial training program, and that says a lot right there, but it's basically to tell these mostly academics that yes, you should think about your projects as being a startup company. And so this I-Corps, it was all virtual, and I don't know if this was post-pandemic thing or if they used to drag everybody to Washington to do the I-Corps. I'd be amazed, because there's 19 roughly projects, and there's 5 to 10 people in each project. So however, for the I-Corps, they limit it to be 5 to 6 participants from each team. And so they basically had us attend classes, do homework. As I said, think about your project in terms of a startup. So you have to think about who are your stakeholders. And quite honestly, until I went through this I-Corps, I never associated all of the open source software development that I've done over the years with stakeholders. But it was a good exercise. So who are stakeholders? It's primarily users, at least in my mind. That's who I tend to think of. But it's also the developers, and it's also any funding possibilities that you may have for your open source. These other bullets, again, they want you to think in these terms. So you have to come up with an elevator pitch. You have to develop this so-called business model canvas. And yes, you have to learn yet another online reporting system called Airtable that the I-Corps uses. Now, normally the I-Corps program lasts, I think it was 7 weeks, and you are required to do 100 interviews of your stakeholders. However, since they didn't decide that we would be going through this I-Corps until December, they got significant feedback from the awardees, and then they rethought things and said, okay, you can, we're just going to have a 4-week effort, and you only have to do 60 interviews. Well, only 60 is quite a lot, as you can imagine, just organizing them, taking the time out of our day, but their days as well to do these interviews. However, after it was all said and done, my personal takeaway was, yes, it was a heck of a lot of effort, but I would encourage any of you to do the I-Corps if you do get a chance. In my opinion, just learning things from these interviews were worth the effort. So that's enough about that, the I-Corps. You can ask questions at the end if we have time. Let's dive right into some of these modeling approaches, which most likely all of you are already aware of, but for the sake of review, my go-to paper is this Chased group paper out of from 2017. So it's Osborne and others. And there they basically reviewed these 5 different framework approaches or modeling approaches. Now they only use Chased in, coming up with these simulations using these different approaches. But it's a wonderful paper. I guess it's sort of considered a classic by now, I'm not sure. And one of their figures is this that we see here to the left. And so, cellular automata is one of the 5. Cellular pots, of course, and that's the one that we will be focused on with CompuCell. What they call overlapping spheres, and we tend to call center-based, is what we do in PhysiCell. And by the way, my primary effort for the last 5 to 6 years has been helping support the PhysiCell project. However, about 14 years ago, I was involved in the CompuCell project. So I've had one foot in both frameworks, you might say. The 4th one there is the Voronoi tessellation approach. And then the last one is the vertex method. And this is showing cell sorting. case it's not obvious. And I will be returning to this very model later, but this is just, and indeed, I'll be returning to this 2017 paper a few times during these slides. So one thing that I think we want to see come out of this OpenVT project is to create and then maintain a living spreadsheet of what frameworks are out there and what their features are, what they're capable of doing. And this may sound trivial, but when you put enough thought into what needs to go into this spreadsheet, it's fairly involved. So this is just showing a screenshot from this OpenVT GitHub organization, where I've just slammed together a super simple spreadsheet, and I just filled in some checkboxes for CompuCell and PhysiCell. I guess the idea, the hope is that developers and leaders in other frameworks will indeed join in this effort and contribute to a spreadsheet like this. I'm not saying it's going to live in this GitHub repository forever. I hope we can move it out into a more friendly landing site on a web page, but time will tell. Here is another flavor, if you will, of trying to organize frameworks and their features. This one was from the HAL paper, the Hybrid Automata Library, from Sandy Anderson's group down at Moffitt. This paper came out in 2020, and I've tried to blow up the left column there, which are the features. And across the top of the spreadsheet is just abbreviations of frameworks which they chose to highlight in the paper. But if you look down the features off to the left, you'll see the usual sort of collection of the modeling approaches. And then you see a couple options there for solvers for your PDEs. And then they have some standalone, is it SBML compatible? What kind of viz can it do? Is it parallelized? Does it run on all platforms? And what sort or sorts of programming languages does it use? So that's just one of a few figures that are out there in papers which attempt to sort of categorize modeling frameworks. Another one is one which I copied from a review that came out of the Macklin lab a few years ago. This is by John Metzgar and others. And here it wasn't as a deep dive as the previous spreadsheet, but it basically tries to categorize again what sorts of methods are used in the various frameworks, which we happen to highlight off to the right there. So you can go look at that. Let's see, I skip one. And then this one is again another table from the Chase 2017 paper. And I've just sort of bracketed highlighting cellular pots versus their overlapping spheres or center base. And off to the far left, that column to the far left, they just sort of have sort of high-level examples of what you might want to do with your model. For example, the first row there is adhesion. If your models needs to demonstrate or have adhesion as part of it, then, according to them in their table, then cellular pots is highly appropriate. And I think in particular, they had in mind when they talk about adhesion, they were really thinking about cell sorting, because under overlapping spheres, you'll notice in that column, they say it's less appropriate to use overlapping spheres for adhesion because there's not as a natural approach to doing differential adhesion. So it lacks doing the total engulfment of the different cell types, which is what I showed in that earlier figure. So this is just yet another table which tries to organize approaches to modeling and examples of models. So sort of the elephant in the room is, what exactly is the difference between cellular pots and a center-based approach to modeling? Again, some of these are obvious to you, maybe all of you, but if not, let me quickly go through some of them. So for cellular pots, the world is a lattice. You have pixels in a lattice, those become the cells. And by the way, I'll have visuals next, so don't get hung up on the words here. But for center-based modeling approaches, you have a center of your cell and a volume. So it's lattice-free. Cellular pots to the left, cellular pots has this idea of minimizing the total energy of your model. So it involves the Hamiltonian, again, as most of you know. In center-based, we just have a classic physics-based dynamics. And in PhysiCell in particular, we have potential functions for handling adhesion and repulsion, but we also have built-in spring mechanics if you choose to use those. In pots, we have no explicit time. It uses Monte Carlo steps. In center-based or PhysiCell, we do have explicit time. Similar situation for spatial units. We have pixels in pots. We have actual spatial units, microns in PhysiCell. And then there's that medium cell type, which exists in pots. So here's a visual. But again, probably most of you know this. Cellular pots on the left is just showing 3 cells. And they're delineated by their cell IDs. So there's one cell here in red, there's a second, and then here's the third cell. And then we show, just a schematic of what that situation would look like in center-based. So you've got your 3 cells. Just a simple visual. So let's get right into standards, I guess. So what might we standardize in this world? Let's start by talking about initial conditions of models, because if we're going to be comparing results of models between frameworks, it would be nice if we could share something resembling the actual initial conditions of a model between frameworks. So how might we do that? Well, the first thing I'd like to say is I do not have any illusions of convincing frameworks, framework developers, leaders, that they will adopt a single standard data format, because they've got years invested in doing things the way they do them, and that includes existing data formats. So the next slide, I'll show you my proposal. But basically, I just want to throw out the idea of maybe we can agree to have an intermediate data format that we can both convert to and then convert from. And so the next part of this slide is simply highlighting, what type of data do we even have for these models? And, no big surprises here. It's all numeric data. We have cell types for cells, and we have either centroids or we have lattice points, depending on the modeling framework again. We most likely have scalar fields, we might have vector fields, we might have tensors. Basically, we need to sit down and figure out what each framework expects in the data that they generate. And similarly for substrates, you most commonly have a standard mesh, rectilinear mesh, and concentrations. But for the most part, I think these slides will focus on cells. So this is the simple idea that I wanted to throw out to each framework about. Each framework would write a script or whatever to convert their output format. Again, let's just talk about cells initial conditions. So they would write a script to convert their cells data format into some standard format. And similarly, they would write a different script which would convert another framework's data, which again would be converted into this common quote, quote, standard format, and it would be able to convert it into their format, their native format. Now, what the heck does that even mean? Well, first of all, what are these different native formats that we're talking about? And I'll just highlight CompuCells and PhysiCell. So CompuCell uses this thing called a PIF format, and it's POTS initial file, and basically, it's just an ASCII file where you give it a cell number, cell type, and then this region of pixels or voxels, if you're in 3D, of where that cell lies. So obviously and so since these are specifying regions, it's going to be rectangular in 2D, x1 to x2. So to complete defining this cell ID number 3, you would need these 2 additional pixels outside of that rectangle. Hopefully I'm making sense here. And obviously you can do that with just 2 more lines in the PIF file. So that's that one. And PhysiCells is much simpler in the sense that it just uses comma separated values to define cells. So in particular, you have XYZ coordinates, you have a cell type, and then you have any additional data associated with that cell. Everything about it that defines it. You can provide there. If you do not provide it, then it will grab that information from other configuration files. So those are 2 example native formats. And then again, I think I'm reiterating what I had before. Basically, you need to be concrete about what sorts of data you're dealing with, and I won't belabor that. But eventually, we need to come to agreement, hopefully, on what sort of standard file format might we use for this intermediate file. And there are 2 that I list here, and that would be a VTK, the Visualization Toolkit. They have an unstructured grid format, which is very, very general. And I went with that in part because I've been aware of and used VTK's formats for 20 plus years, I guess, so I'm quite familiar with it. Another one that was thrown out as a possibility is data formats from Blender, which is a very nice modeling package on its own, very computer graphicsy. So personally, I don't have a dog in this fight, as they say. However, initially I have gone with VTK just to demonstrate how this might play out. So here's a simple example again of we have a PhysiCell initial conditions of cells on the left here and just 2 cell types. And when you display those in PhysiCell Studio, that's what it could look like with this hexagonal packing. And what I did was I wrote a Python script which converts these cells, center-based, into a VTK format, the VTU. And then I wrote another script, which you could consider a CompuCell Python. These are all Python scripts, by the way. I'm a big Python person. And it could convert this VTU file into a PIF file. And after you do that, you get what would be displayed there on the right. Never mind the colors, those can be changed after the fact. But the fact that it comes out looking reasonably close was what I wanted to demonstrate. Now I have an arrow here that it can indeed be trickier going the opposite direction. Why is that? Well, because CompuCell cells can have morphology, right? They can be these twisted things. And so, and even though they have a center of mass, when you convert it to a center base, it just may not be exactly what you were looking for. So again, we need to look at this more closely. And looking at my time, I see that I'm not sure exactly. I guess I'll just go until somebody speaks up and says, I've got maybe 5 minutes left. Hopefully one of the organizers will do that for me. The second thing that I wanted to highlight about what we might standardize is the idea of a cell's phenotype. And I'm just going to dive right into it and perhaps speed things up a bit. So multi-cell DS is this thing which probably some of you listening today, watching, were co-authors on. This came out of Paul Macklin's efforts primarily, and Sam Friedman's back in 2016. They have this preprint. And the idea was to come up with a data format for biological cells and experiments, which could capture all relevant information. I know it sounds like a stretch, but you can go read that preprint yourself sometime if you're interested. Basically, it's just an attempt to come up with a standard. And one of the core ideas in this multi-cell DS was to have a clearly defined phenotype for each cell type. And I'll say more about that next. But that is one of the standards that would be lovely to see come out of this. OpenVT, again, time will tell. But what do we mean by phenotype? Well, it's probably things that you already think about as being related to a cell's phenotype. Basically, the cell cycle is part of a cell's phenotype. And here down on the lower left, we have the XML snapshot of a PhysiCell configuration file. So it's model, basically. So you have a cell definition, and then you have this phenotype block. And what we're looking up here is the PhysiCell Studio rendition of what the XML has. So perhaps this is friendlier on the eyes looking up at the Studio. But for each cell type, we have these subtabs, and those capture parameters for the phenotype of that subtype, of that cell type. And so, as you might guess, it can get, I won't say cumbersome, but it's involved to prescribe all of the relevant parameters for these various phenotypic sub tabs here. So again, I won't belabor this, but you can go read the PhysiCell Studio preprint. We were hoping to have it actually published by today, but didn't quite make it. So stay tuned for the actual publication. Now, does this phenotype idea have any traction at all? It turns out that it has some traction because a PhD student of James Glazier's, Giuliano, actually wrote this other preprint, and he called it PhenoCellPy. And the basic idea was, can we take the phenotype idea from PhysiCell and port it to CompuCell? And he did this to some degree, and he's got a repository out there. But it does rely on having your framework have a Python API basically to it. So ways of extending, either defining or extending your model with Python, because his interface is all through Python. And he demonstrated this, that he could do this port over to CompuCell just for the cell cycle at least. So the question is, can we extend it to, well, a couple of questions. Can we extend it to other concepts other than the cell cycle in the phenotype? But the other question is, can we extend it to other frameworks that do not have a Python interface? The other thing that Giuliano did was actually attempt to convert a PhysiCell model, that is the XML, the configuration file, into a CompuCell model. So can you convert a PhysiCell XML model into the files that are needed to run a CompuCell simulation? Again, he did this, at least in part, and got a good starting place, I think, for the rest of us. I do like the comments that he left in his conversion scripts, which I may actually adopt. Some of this is left as an exercise to the reader. I wouldn't have thought of including that in some of my scripts, but I may adopt that. And he demonstrates this. Well, I demonstrated it after I forked his repo and then had to make a few edits. I probably based on the latest configuration file. So the latest version of our configuration files in PhysiCell. But basically, again, just a visual here. The bottom line here is the converted model actually runs when you run this conversion script. So it takes the PhysiCell model, it generates the files necessary for a CompuCell model, and you can actually run it. And I need to dig into the details of to make it look more like what I was expecting to see. And I left off the fact that this is one of the sample projects in PhysiCell called BioRobots, where you have worker agents drag cargo agents to director agents. It's a cute little model that comes bundled with PhysiCell. But anyway, Giuliano has taken these initial steps for porting these things between these two frameworks at least. Okay, now I'm going to move over into reference models. So what time are we looking at here? 22. So the OpenVT, we've been trying to contact lots of other framework developers, get them engaged, encouraging them to become part of this community. And as part of that effort, we had a meeting last month where we discussed, and then I think came to agreement on these 5 reference models. So the simple idea, as you might guess, is to have each framework go off, and I think the way we left it is there are teams. So we split out into teams that would focus on either 1 or 2 reference models. And then they would come back and demonstrate how they could do that in whatever framework they were using. And I'm not going to talk about each of these, but the first one is fairly straightforward, I think. Can your framework handle a random walk of cells? And can it handle a biased random walk? Can you demonstrate monolayer growth, sort of leading up to wound healing, perhaps? Number 3 sounds super simple: 1 cell that chemotaxes just across the domain. And number 4 is cell sorting. I'm in that group. And the last one is angiogenesis. So again, I'm not going to touch on each of these, but I am going to touch on a couple of them, number 3 and number 4. So in this one, thanks to Michael Getz for putting this together. This is CompuCell, and it's doing just what, am I not able to, let's see, I need to undo that. Here we go. So yes, here's a single cell, a CompuCell cell that's chemotaxing across this gradient, which we see down below. And this bottom graph is simply showing the location of the center of mass. So the blue is the Y. So it basically never, let's see, is that right? Yes, the Y-axis. So it basically never leaves, Y equals 0. And then the red, of course, is the I'm sorry, not axis, but coordinate. So the red is, and so it does indeed chemotax. So I guess we can check that one off, whoever had that model in their group. And then similarly, here's PhysiCell doing something similar, and notice it's not as blobby as the PhysiCell cell, of course. But the other thing I wanted to point out is down here as part of the phenotype subtab for motility of this particular cell type, we have these parameters which affect the speed and the persistence time of a cell, and also the migration bias. So for whatever reason, I chose it to be extremely biased. So that was sort of a cute little visual to show how these reference models might play out. And obviously, I sidestepped the question of reproducibility for these for the time being. Yeah. And then the cell sorting, one of the other reference models, is more challenging, as you might guess. Can these frameworks demonstrate cell sorting just using differential cell adhesion? And again, just cropping out 2 rows of that earlier figure from the Chase 2017 paper. This is the end result. So here's the starting initial conditions. And then over time, right, these do their sorting thing, and then cellular pots comes, the final result is this total engulfment. For overlapping spheres or center-based frameworks, you may not see that. And this is a fairly deep question of why is that? What are the reasons? Is it possible if you go to enough effort? So all of these interesting questions will come up. I'm getting near the end here. One slide on can we think about it incorporating these tools which allow for, putting more modular functionality into the overall modeling workflow? If so, how much work will be involved in doing that, and what sort of performance hit, if any, one would take? So I throw out this example of, well, it would be nice to demonstrate anisotropic diffusion solvers for some of these frameworks. So if there's a solver out there, then can we plug it in to one of these modular integrative workflows? And I only list a couple of these, as these have been raised in various workshops that I've attended related to multicellular modeling. One is Vivarium, and the other is this Muscle3 platform. I think the Muscle3 person presented at this same working group series a few, well, it was back in February, I think. So I'm just going to leave those references there and leave it as an open question about experimenting with these for OpenVT. The last topic is reproducibility, which I will not have time to give due credit to, but can we reproduce, quote, quote, model results across frameworks. And to do that, you have to be able to quantify what are the results that you're getting. And how does one do that? Well, the usual approach is either statistical measures. It could be, since we're dealing with spatial models here, it could to some degree be geometric or topological. And I raise the question, are we talking about reproducibility just at the end of a simulation, you know, whatever the end is, or do we have to be concerned about the entire dynamic trajectory of a simulation? Lord help us if we do. And then this last bullet is a very recent thing that I became aware of at the conference in London last month, in the breakout session there, which TJ was in charge of, and that's the stochastic simulation reproducibility. So I'm unfortunately not going to have time to say more about that, but there's a GitHub repository you can go check out. And then one final thing which I've been playing with recently is doing an image processing approach to PhysiCell results. Can we take the final results of a cell sorting simulation, convert it to grayscale, do a histogram of where do those pixels lie in terms of their intensities, and then can we do a contouring to say how many clusters appear in those final results? And it's a work in progress is all I can say. But I will mention Jim Sluka's talk a couple of weeks ago where it may be somewhat related to this very concept. Future plans. We will have a landing page very soon, I hope. And there we will have lots and lots of additional things. I'm going to stop there for now and at least have a few minutes for discussion, I hope.
I have one if no one else has any.
Go ahead, Jim.
Randy, do you think that phenotype specification tool could be used more generally? A lot of times, I mean, in the CompuCell classes, and you probably do it in the PhysiCell classes as well. One of the first things you try to get a young modeler to do is just to sit down and make a list of, these are the cell types in the model. These are the secreted fields in the model. These cells can grow and these cells can die. So can that little tool be used kind of standalone?
Well, that would be probably wishful thinking, but I would love to attempt to do just that, Jim, with you. Quite honestly, it's still very fresh for me. Just sort of when I volunteered to do this talk is when I got more serious about looking at it. But yes, I would love to follow up with you about looking into that.
Okay, yeah. I know maybe we could create a web interface or something.
Even better.
So that, right, so that somebody doesn't have to download anything at all. And the only tricky part is how you reload back in if somebody wants to edit it. But that might be an easy way to also include things like, if you're going to assign a cell as epithelial, can you also include a link to Go or cell ontology or something like that? Okay, cool. We'll talk. Thank you.
Thank you, Jim. DJ.
Thanks, Tom. Hi, Randy. Great talk. So I work with a couple of different platforms that use different methods, like you with your background. Excuse me. I was wondering if you could comment a little bit more on some of the nuances and subtleties concerning reproducibility between, in particular, between platforms that use different methods. And a simple example that comes to mind is the difference in how cells are represented using a center model versus a cellular POTS model, and that you have implicit versus explicit cell shapes. And maybe some metrics of quantifying, say, cellular configurations directly translate between the two, but there are a lot of things about those different formalisms that are not obviously comparable. And so in terms of reproducing results What can we feasibly and reasonably expect?
Super loaded question. And in some sense, I feel like I should flip it and hand it right back to you, since you have been thinking about reproducibility. So I alluded to your effort there with stochastic simulation and reproducibility. I don't know, TJ. Part of me wants to say that we want to, well, it's going to be a multi-pronged approach. How's that for trying to step away from, but part of it, I want to be sort of higher level. What does your final result even look like? And let's just keep it simple in 2D. Let's not even go to that crazy third dimension. But just what are the structures that let's start with human vision. What do you see as a human when you look at the final results of a simulation? And I think it's best to start with simpler cases like models and simulations, which do indeed end up in some sort of structures, coherent structures. I guess I'm trying to distinguish that from, say, an immunological model, even though there you could also have structures, but maybe less so. But yeah, trying to define what are the coherent structures, and surely in other scientific fields, this has been touched on, right? I want to say just broadly computational geometry, but surely other fields about extracting structures from data and then saying between frameworks, okay, well, my simulation shows these 5 structures, broad structures, and let's just start with that, the number of structures. But yeah, I mean, there's the geometric and spatial structures. There's, then it gets trickier if you've got topology going on. Then that adds another level of difficulty. Maybe I'm totally missing the boat, and maybe it can boil down to, I won't say simpler, but statistics. And here's where I think you should chime in down the road, if not today, with what you've been doing. But even there with your work, isn't it the case that we need to define what it is we mean by a metric for these simulations?
Well, that is my opinion, yes.
Yeah. Thanks for doing that sanity check for me.
Yeah. Well, and I don't want to steal your thunder. I just, there's, I mean, between my experience with working with the simulation platforms and then with quantifying stochasticity, and I want to at least give a little bit of room for you to give your thoughts on that, because I mean, your participation in London and whatnot, I know that you have a lot of background and a lot of opinions on this as well, and so I wanted to make sure that gets a little bit of screen time and isn't lost. All the other great content that you discussed today. So thank you very much.
Lutz.
Randy, thank you. If I may, so again, to the side topic that you mentioned in the very beginning with the iCorps program. So is it logical that you can turn these open source projects that we all run into some commercial flavor? What is the motivation for enrolling the 19 open source projects into an iCorps project? I think one could have like an extra consulting track maybe alongside with the open source software to teach others and maybe get them started with the project, or what is the commercial aspect to it, to an open source project?
I think you should have been in attendance at the iCorps, Lutz. It sounds like you've got some ideas there about consulting. Quite honestly, I came into this with what on earth are we even talking about? Because, I've got nearly 30 years of doing open source software development. And so making any money off of this is sort of the last thing in my mind. It's just sort of it's academia. We write open source software, we make it available, we get funding from whatever national institute. But certainly people have, so Kitware in particular, the creators of VTK and many other things, they are an open source company. They started in the 90s, I think, when they came out with VTK, and they are very, very successful about, a commercial, for-profit company. I think that's fair to say, based on open source software. So it may sound unusual to you if you've lived in the academic space, but yeah, certainly there's various avenues, and I think Kyle may want to chime in perhaps with an answer or possible answers. Kyle?
Well, we're kind of at the end, but I think we're still trying to figure out what we want to do. But what we think the opportunity is that for a long time, this area was kind of following the crafts model, where we had single labs developing packages and solving similar problems over and over again on their own. And kind of like, there was a time when clockmakers all made their own gears, and they were all custom fit to each clock and had the right tolerances and everything. And so I think we're moving into a time where reproducibility, validation, calibration is becoming more important to people like the FDA, like the NIH, like the EPA, and using these things as alternatives to animal testings or supplements that can maybe be more reproducible across models or interchangeable at the very least are kind of big goals. And so we want to, start with some reference models and kind of bring some of those lessons learned across labs, where maybe one lab has a really good way of doing something that's reproducible, and others may learn from that, implement their own version. So, there was a time when everybody made their own nuts and bolts, right? And we don't, then there became a standard around that, and then things were interchangeable. So I'm not sure what the right metaphor is for this industry, but I think together we could identify subcomponents or things about data, best practices that help us kind of advance the industry and make us more productive. So that's kind of part of the notion, but we actually want to welcome your participation to kind of come think this through, help us, and I think that's where we are right now.
And I fully agree. There's exactly this work to be done, but I think once it's done and we want to have it published OpenSS and be part of the open source code base, then I think there's no commercial follow-up on it. I think the work has to be done, that's true. And I'm not sure what the ongoing effort would be that one can then sell. There is an initial effort that needs to be done, but we want to have this then part of the open source code as well. So I'm not sure exactly what one can do months after months and sell it again.
Sorry.
Sorry, no good answer, Lutz.
Yeah.
Well, yeah, that I'm here, I will comment that probably this effort we have discussed from the computational side, being myself an experimentalist, I think we should do the same efforts on that side. So standardizing measure so that we can feed these models with things that we all know what they mean and they are kind of agreed upon. And that's more a comment than a question, I guess. But on this note, I think we have very good discussion tonight. If there are any last questions, I'm happy to, I'm sure we are happy to listen to. No? So on that note, I will just close tonight's seminar. Thank you very much, Randy, for coming. Thank you all for the nice discussion and for coming up today. And I guess I will see all of you in a couple of weeks.